Hello Mindsetters, welcome to today's Grade 12 Life Sciences show, proudly brought to you by Macmillan. I'm Katleho. Today we have a special revision lesson on genetics and evolution. We have selected highlights from lessons shown earlier on in the term just to help you guys revise all the work we've done together in the first term. You can download the notes for today's show from learnextra.co.za/live. Also, guys, please post all your comments and questions on our Facebook page, which is facebook.com/learnextra. Also on our Twitter handle, which is @learnextra, so that we can help you guys with all your revision. Guys, let's get on with your lesson today.
All right, we move on. By carrying out these experiments, what Mendel did with the plants, he determined that two alleles for each character segregate during gamete formation. He didn't know that this was meiosis that caused this. Anaphase, when they separated, they moved apart. And this called the law of segregation. I think at that point, Indy, it's in between a definition. The definition is done. Whatever follows, we'll explain after the break.
Fantastic. Guys, what I'm actually going to do is I'm going to post that
Lots of that. Remember, mono-hybrid cross is what we are doing at the moment. Mono, one. Hybrid cross, taking one set of characteristics at a time and crossing and looking at the probability. Fortunately for you, you don't do the second one, which is called dihybrid. That means we take two characteristics. Imagine taking a tall plant that has round seeds with a short one with wrinkled seeds. When we do that, obviously we're looking at two characteristics at the same time. It's called dihybrid. You don't have to worry about that. It's out of your syllabus. We look at monohybrid. So the example we looked at above level was the monohybrid cross. We looked at one characteristic at a time. What we did with the balloons was also the same thing. We are looking at one characteristic at the same time. Modern interpretation of the law of segregation says, remember, Mendel said the two factors segregate. What do we say now after we've learned? The characteristics of an organism are controlled by a pair of alleles. We already spoke about this in DNA, that for every characteristic, there is a pair of genes. These pair of genes are called alleles, okay? And these alleles separate into different gametes as a result of meiosis. Remember, anaphase, the chromosomes move to opposite poles. Anaphase 2, the chromatids move to opposite poles. So when they became gametes, so the two factors or the two genes were carried on these chromosomes, and they were separated so that each gene each sex cell or sperm or egg carries only one of the two. So they have separated. So the law of segregation states that for every characteristic, there are two genes or a pair of alleles. These alleles separate during meiosis. As simple as that. Law of segregation. We're still talking about monohybrid, and the one that we have discussed before of the tall and the short. Obviously, one was dominant, one was recessive. And this shows complete dominance, when one completely dominates the other. So when two individuals with pure breeding, contrasting characteristics. Contrasting means one is dominant and one is strong in this case. The F1 generation all display the dominant characteristic. So this is complete dominance. Because why I did that now? Because the next one we are talking about, not all the characteristics follow in this pattern. Some show incomplete without the pause now. Incomplete, not incomplete. This is incomplete dominance. Now here what happens? Neither allele is completely dominant over the other. For example, if we take a red balloon, if we take a red, well, let's call it a red flower in this case, and we take this red flower and we cross it with a yellow flower, and the resultant offspring are all orange. So what has happened? Neither of the two characteristics were dominant over each other, and therefore none of them show in the offspring, in the immediate offspring. We get an intermediate color. So red and yellow gave us orange. In nature, the example that's on the board, very simple. We're taking snapdragons as flowers. White flowers and red flowers give us the pink variety there. So neither one of the two is dominant over the other. So they're not dominant at all. All right? We go one step further. I'm going to go back to how we're going to work it out just now. Others display co-dominance. So we had complete dominance, we had incomplete dominance, now we are talking about co-dominance, where genes have alleles that are both expressed in the heterozygote individuals. An example, you see this in a book, you have your reddish cow with the white cow, and you get roan. Roan is a color which has reddish hair and white patches on it. So that means both the red and the white show up in the offspring. Now, in our example of our red and yellow flowers, if we mate them and we get this following result,
Look at that.
so we have a yellow flower with red spots. Or we could have had a red flower with yellow spots. So both characteristics are showing up in the offspring. So in this case, we are saying that this is co-dominant. They're sharing the dominance. Are we clear with this, guys? You can't forget this now because you're seeing the color in front of you. Now, let's go back to our incomplete dominance. We gave the example. Now we want to say, how does it work? So we said that the father, not the father yet, the male flower and the female flower, if the male flower was red and the female flower was white, so we have given the phenotype. Notice I am showing you here as well the format of how to answer this question. The genotype, if we're saying we're starting with homozygous, so that will be capital R. And notice, normally, we would have shown small r because of our complete dominance. We move away from this because they're neither dominant over the other, so we use capital W's. You have to use capital letters to show that there is no dominance between these two. So there's your genotype coming out there, and meiosis takes place, and the gametes would be R, R, W, W, separated. All we're saying is that meiosis separates these guys. And when we use a Punnett square, we write the word fertilization here. Here we will put R, R, W, W, R, W, R, W, and so on. They're all going to be RW. And therefore, our individual will be 100% hetero and 100% pink. They'll all be pink. If we take the pink variety and we mate them, then we will get the ratio of 1:2:1 in the phenotype generation as well. We get a 1:2:1 and 1:2:1. 1:2:1 genotype, homozygous, heterozygous, homozygous, and red, pink, white. That's what we will get in our result of this as well. Now, in the next one, we also ask you to do the same thing here. So this was red and white. Again, we can use the same letters, R, R, W, W. I'm not going to do the gametes because you've already learned how to do that. So we have the same thing here, but look at what happens because this is co-dominance. The genotype remains the same. They will all be RW, heterozygous. But the phenotype would be this color called roan, or we would say red flowers with white spots, if we're talking about flowers. Or if we take cows that are white and black, we get white cows with black spots, or black spots with white cows. Black cows. Black cows with white spots. Indy, you're not helping me here. You're
I'm just
laughing at
watching.
me.
I'm watching you. It's very funny.
Okay. Notice,
How does
guys, look
it
at the
feel?
mark allocation. Look at the mark allocation. You're getting a mark for giving the phenotype. I mean, the phenotype there. Sorry, there we should have actually. Sorry. Good that we went back there. We wrote the wrong thing in the wrong spot there. Good. Lucky we went there. Guys, phenotype. It's red and white. Remember, phenotype refers to the for pheno features. And here then we would say genotype would be there. So watch what you're doing. You're getting a mark if this whole sentence is, well, you can't call it a sentence, but this whole line is correct. And you're getting a mark if this whole line is correct. So you're getting one there, one there. You're getting, if your gametes are all correct, R, W, you get another mark there. For the Punnett square, you get another mark there. And watch what they say at the bottom. For writing P1 and F1, or parents and offspring, you get a mark. And for writing meiosis and fertilization, both these things must be written in the correct position. If it's not in the right place, obviously, if I can just go and write meiosis and fertilization anyway, they're not going to get marks for that. You've got to put it where it happens. And where does it happen? In the formation of the gametes, meiosis will come here. And fertilization would come after the gametes have been formed, in line with the Punnett square. Keep this in mind and follow for every genetic problem. Follow this pattern. Otherwise, you will lose marks unnecessarily. What a better way to start, Indy, than a lovely picture.
Is it a boy or a girl?
That's the
What
question.
could it be?
That's the question. So, sorry to disappoint you, you young ones, when I said how sex is determined. When I spoke about how sex is determined, you had a whole lot of other ideas in their mind, indeed.
They were thinking other
They
things.
were thinking heavy
other
stuff.
things.
No, when we say how sex is determined, we talking about how the child that is about to be born, how is it determined for that child to be a boy or a girl? And so there we have the mom there with a nice round tummy, and she wants to know whether she's having a baby boy or a baby girl. And how is this done? Now, remember last week, we went through the whole procedure of P1, genotype, phenotype, etc. And then we went to the last part, and that was the Punnett square. We also spoke about, when we spoke about meiosis, we said that the male has the sex chromosomes XY, and the female has the sex chromosomes XX. And that's what helps us to determine the sex of a baby. So here's the father, XY, these are the gametes. So he can either have a sperm with X gene or a sperm with Y gene, or we call it the X chromosome and the Y chromosome. The mother, on the other hand, does not have this choice. She has only X and another X. So the females can only offer the X chromosome. The males, on the other hand, can either donate or contribute, I would say rather, contribute the X chromosome or the X gene or the Y gene. So if we put this on a Punnett square, that gives you XX, that and that gives you XX again, YX gives you XY or YX. Remember, you can write it either way, XY. So XX is a female, and XY is male. So according to this, guys, what are the ratios that we are looking at every time fertilization takes place? What are the probable, note the word, the probable ratios? The probable ratios according to this is obviously 2:2, or rather made simpler, 1:1. Or we can say 50% is to 50%. That means every time fertilization takes place, according to the laws of probability and according to the laws of genetics, the probability is 50% chance of having a boy child and 50% chance of having a female child.
Okay, guys, I hope you're really enjoying this revision session. If you are struggling and need help, remember to post on the page or send an email to helpdesk@learnextra.co.za. Now it's time for a break, so don't go away. We'll be right back. Welcome back from the break. Please remember that this month is Library Month. We really think that reading is important and can be a whole lot of fun too. So remember to join in the chat on the page and tell us about the books you love. Look out for our book competitions too during Easter revision. We are going to have a whole lot of fun. But for now, let's get back to revision. Over to you.
Who contributes? Who determines then? Besides God. Besides God, we're not talking about God now. From the couple, the married couple, husband and wife. Who determines whether the child is going to be born is a male or a female? From what I said earlier.
Oh my gosh.
Is that a tough one in there?
That is a tough one. I mean, I'll probably tell you like
The male must have the Y chromosome. So where's the Y chromosome? Only in the father. So only the father determines whether that child is going to be male or female. Again, to all those very religious people, including myself, we're not talking about God in this equation that we're talking about. We're talking about the probability factor. So the male is the contributor. So James, Abraham, John, all those guys in the background.
Not Belinda,
Not
who's our
Belinda.
director.
Just note that if your wife had a girl, another girl, it's not her fault.
It's his fault.
It's his fault because his Y chromosome is too lazy to do the job. Okay? So the male will determine the maleness. It's only the male that contributes the Y chromosome, so therefore it is the male sperm. It depends which sperm reaches the egg. Remember that. And we spoke about the sperms, the different one of the two genes in each of the sperm. Mendel's law of segregation. Each of the two alleles separate into the different sex cells. So that's what we would do. Now remember, in exams, they like to ask this question. Aslam and Indy had 5 children. And the first one was a boy, the second one was a girl, the third one was a girl, the fourth one was a boy, the fifth one is a girl. What is the probability of them having a girl child in the 7th instance? The answer will still be
80%.
50%. Because the probability is still 1:1. Doesn't matter. Just understand this very carefully. When I was in grade 12, Indy, I always thought, not in this particular problem, but I always thought this would be the first child, that's the second child, that's the third child, that's the fourth child in the Punnett square. What this actually means, that every time fertilization takes place, these are the probabilities. It doesn't mean that's number 1, number 2, number 3. You could, and we know for a fact, there are people that have only boy children. There are other couples that have only girl children. So where does this go? This only gives the probability. Remember that. Okay? So remember, whenever you see that question, what is the probability of them getting a boy or a girl? 50-50. It doesn't change, no matter how many children they've had before that. Good. We move on to something a little bit more complicated, but if you listen carefully, and if you use and apply your mind, it should be easy. We're talking about sex-linked traits. Again, don't raise those eyebrows. We are talking about those genes that are on the sex chromosomes. That means a gene that codes for a particular characteristic indeed, which is found on the X or the Y chromosome. Remember, we have 23 sets of chromosomes. 22 of those are autosomes. That means they are normal body chromosomes. And the last pair, pair number 23, are known in a scientific term gonosomes or gonadosomes. That means the sex chromosomes. Those are the ones that determine the sex of the resultant zygote. In addition to this function of determining sex, they may also carry some other characteristics, or rather, they may carry some other genes which code for other characteristics. Those genes that are carried on the sex chromosome are called sex-linked genes. Yup, I nearly fell over the board there, Indy. You didn't see that, luckily.
I would have
saved me.
I would have saved you. I would have
I
saved
think you wouldn't have
yeah.
made it through there, Indy. Okay. Certain human disorders, like hemophilia, are caused by sex-linked genes. That means the gene that causes hemophilia is carried on the sex chromosome. In this particular case, on the X chromosome. The Y chromosome is much smaller, so therefore chances of it carrying many genes is reduced. It does carry, but much less. And we are more interested in the ones that are carried on the X chromosome. So there we have already explained that whole story there. If a male inherits the recessive allele on an X chromosome from his mother, obviously he will receive the X chromosome from his mother. If we go back, back again. Sorry, no, forward. There we go. Now, remember, here's a male. XY. Where did he get the Y from? From the father. There's the Y there, from the father. This X he got from the mother. That's what you've got to keep in your mind as we talk. So, if a male inherits the recessive allele on an X chromosome from his mother, he will have hemophilia. If his mother had hemophilia and she transferred, or if she was a carrier even, and she transferred this X chromosome to him, then he would have hemophilia. Why, you ask? Because it's recessive. How come? And I said last week that for the recessive characteristic to show, it must be homozygous. But let's take this case. He has received one X with a hemophilia, small h there, superscript, and he has Y. I said earlier that the Y chromosome does not carry the hemophilia gene, so it doesn't even carry the normal gene. So because he doesn't have another gene to counteract this one, even though he has it only one, that means he has it on the X chromosome, he has hemophilia. Whereas a female, if she inherits one from the mother or the father, and the other one is the normal one, she has the normal, the cat is there, so the mice can't play. Remember, when the cat is away, the mice can't play. Even though this is not homozygous, but there is no cat here by the Y. Y can't carry the cat on its back, if you want to put it that way. So therefore, the boy gets hemophilia in that case. We'll look at some examples a little later. If a female inherits the recessive allele, she would not have the disorder as long as the other X chromosome carried normal dominant allele of the gene, as I showed you in the example there. She had the normal one. However, if she had two recessive ones, then obviously there's no cat there, so the mice will play, so she'll be hemophiliac. Hemophilia is just one example. Color blindness, red-green color blindness, is another example of sex-linked disorders. Let's move on. And there we have a table to show what we are talking about. Look at the genotype. XH, capital HY, that means that's a normal gene. So he has normal blood clotting time. Okay, let's just go back again. What is hemophilia? Hemophilia is a condition where the factor that is necessary to cause the blood to clot when you are bleeding, when you have an injury and you get a cut and it starts bleeding, the blood clots. There's a factor that causes that. If that factor is absent, you would be suffering from hemophilia. So the person that has the recessive gene there, in this case here, will have hemophilia. In this case, XX, capital, homozygous, normal. XH, heterozygous female, she's normal, but she carries the recessive allele, so we call her a carrier. She's a carrier. She has the potential of passing this on to her sons especially. Okay? If she has to pass it on to her daughters, then her husband must also carry that gene, so that X small h would make the daughters also hemophiliac. There we have this last scenario there. So these are obviously examples of mutations. Mutation means something going wrong. And here's a classic example. Something's gone wrong. This person cannot clot their blood normally. Other mutations are autosomal. This is a sex-linked one. The others are autosomal, meaning they affect the other chromosomes, the other 22 pairs. And some are recessive, while others are dominant. Okay? We are going to do examples, so don't stress today. I'm trying to push the content and then do questions because we've been falling behind on our question time. So we're going to do questions, and when we do the questions, then the way you answer it and the format will become clearer, okay? So don't stress about that. We're now looking at a pedigree diagram. What is this pedigree diagram that everybody's talking about? It is a chart or a diagram to show graphically the inheritance patterns over a number of generations. Couple of things that we need to know here. Obviously, they are usually used to study the inheritance of genetic disorders, but they can be used for anything. Pedigree diagram can be used to show a simple monohybrid cross as well. A pedigree diagram can be used for anything you want to use it for. It is mainly used by genetic counselors to map out to a potential couple that, look, these are the scenarios in front of you. For example, a particular female has hemophilia, or rather she knows that in her family there was hemophilia. She wants to get married, and the couple are now interested to see whether they're going to have children with hemophilia. So they see a genetic counselor, and he then maps out a pedigree diagram like this and say, look, these are the scenarios that can happen. He cannot tell them, hey, listen, Indy, Aslam, you can't get married because your children may be hemophiliac. All he does is he tells you, look, this is the situation. This is what can happen. After that, Indy and Aslam must decide whether they're going to get married or not.
It's a tough decision.
It's a tough decision.
It's a very tough decision. So what are the risks? What are the percentages? How do we make that decision, Aslam?
This is what we're going to answer when we look at the questions later. And the important thing is, when the counselor gives us these options, and the same with Down syndrome, if you go to a counselor and he wants to know whether you have a risk of having a Down syndrome baby, all he does is he tells you, look, these are the probabilities that you're going to have a baby with Down syndrome. Indy then will have to take the decision, look, this is what Down syndrome is all about. These are the symptoms of Down syndrome. These are the symptoms of hemophilia. These are the symptoms of color blindness. Will I be able to cope with rearing a child like this? So Indy or whoever else will have to make the decision. Okay? And know when you make the decision, any decision, and this is not about life science, this is about life. Any decision you make, you have to take the responsibility of whatever comes after that with the decision.
Wise words.
Okay.
Hey,
Good.
life lessons as well, guys.
Life lessons all
as
the
well.
time. Life science. All right. What we need to know in a pedigree diagram. First of all, you'll notice there are squares and there are circles. There are some that are shaded and there are some that are unshaded. For example, in this particular illustration, we have the circles there, and the key is given to you. Circles are females. Okay? Circles represent females. Squares represent males. So that's the first thing we have to understand. The ones that are shaded usually in a disorder pedigree diagram, the ones that are shaded are the ones that will have the disorder. So we'll ask the questions just now. And the ones that are unshaded are the so-called normal ones or the carriers. They can be carriers also, but normal. Okay? Second thing that we need to know is that there are certain layers. Can you see that there's 1, 2, 3 layers here? These 3 layers represent 3 different generations. This is the first generation, this is the second generation, that is the third generation. Also, we have to note that only when the line interconnects 2 organisms, or 2 individuals rather I should say, and from there another line connects other individuals, like 4, 5, 6, 7. All of these are offspring of 1 and 2. Number 5 is not an offspring of 1 and 2 because it's not connected to 1 and 2. It is only connected to 4. And number 8 is also not their offspring. It is probably the wife of number 7, and this is the husband of number 4, and so on and so on. So a couple of things that you have to remember. Circles, female. Square, male. Shaded, usually the affected organisms or individuals, and unshaded, the normal ones. The different levels, different generations. There must be a connection to be offspring of the one before. And this one here is talking about albinism. That should be an I, albinism. Albinism is not a sex-linked disorder. It is an autosomal disorder. And it is caused by a recessive allele. Now, if we have to complete this particular chart, very simply, remember what I said last week. When the cat is away, the mice will play. Albinism is caused by a recessive disorder. So obviously, if an individual is affected by albinism, it must have homozygous recessive. That is the key in this problem. So what can we do now? We can, and let's say we're using capital A and small a. So we go immediately to the shaded ones and we fill in there small a, 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 small a. So these individuals, number 1, number 8, number 13, number 14, they are all homozygous recessive. Why do we say that? Because albinism is caused by a recessive gene, and a recessive characteristic will only show itself if it is in the homozygous. Remember last week we spoke about a test cross. A test cross is used to determine whether a dominant organism or an organism showing the dominant characteristic is homozygous or heterozygous. For a recessive characteristic, we don't have to do a test because it has to be homozygous. From there, we need to go to the parents now. Let's look at 4, 5, 6, 7. All of them should have a small a. Why? Because the mother had the two small a's. So she contributed one gene to them, all small a's. So they all have the small a. But are they shaded? No, they're not. So they are normal. So they should have the capital A as well. So they are all heterozygous for this condition. So they will be normal, but they will be carriers. We'll come to number 3 just now. Let's look at 7 and 8. 7 and 8, they have these offspring. Obvious, number 8 would have contributed a small a to number 12. At the same time, because it's normal, it should have the capital letter A to make it normal. So that is how you work that one. Now this is where you get a little confusion or trickiness, but it's not a train smash. All of these offspring are showing the normal condition. And the individual 4 has heterozygous for the condition. Now, what about this parent? Because we don't have further evidence, we would then be able to assume that this could be either a or a small a, because we don't have any other suggestions here. And if all these offspring are a, they could have been a, or any one of them could have been a small a, because they could have inherited this small gene from the mother and the other normal gene from the father. Now notice, if you do what I just told you, first fill in the recessive characteristics and thereafter work backwards or forwards or upwards or downwards.
Okay guys, I hope you're really enjoying this revision session. If you are struggling and need help, remember to post on the page or send an email to helpdesk@learnextra.co.za. Now it's time for a break, so don't go away. We'll be right back. Welcome back, Mindsetters. Today we are doing revision. Even though we are selecting highlights from previous shows, you can still follow me on Twitter @LearnExtra or post or comment on our Facebook page, facebook.com/LearnExtra. Right now, let's do some more revision.
Where we go straight into it, a hypothesis. What is a hypothesis? What is it to hypothesize? We are doing it every day. I say to myself, if I leave Azadville at 4:30, I should reach here by 5:30. You notice? If I leave Azadville at this time, I will reach somewhere at a certain time. I did not say, will I reach Randburg at 5:30 if I leave there? That's a question. I didn't say to determine whether I will reach Randburg at 5:30 if I leave Azadville at 4:30. That is an aim. So we say, if I leave, so it's your if then. So it's your cause and effect, an independent variable and a dependent variable situation. So what is it actually then? It is a prediction. So you're seeing something and you're making a prediction of this phenomena, something, a phenomenon or phenomena, plural, which can be tested by observation or experiment. If I say that if I leave Azadville at 4:30, then I should get to Randburg by 5:30, I can test that. Because then I leave Azadville at 4:30, and I see whether I get here on time. At the end, when I reach here, I can then make a conclusion based on the experiment or the observation, my test. So first I made a prediction, the hypothesis, then I carry out this investigation, experiment, I do the thing, and at the end I can say, yes, my hypothesis was true, and if it was true, we accept the hypothesis. If it was wrong, then we reject the hypothesis and we start again. So it is not yet a fact, it's not even a theory yet, it's merely your gut feeling. But you need to take a stand. You can't sit on a fence. You've got to say, this is going to happen or that is going to happen. So that is what a hypothesis is about. Now, after a hypothesis or hypotheses are tested, reviewed by scientists, not only in Johannesburg, not only in Gauteng, not only in South Africa, but globally, then we can then accept this hypothesis, and the hypothesis can be converted into a theory. So what is the theory then? It's an explanation of a group of physical phenomena, some of the things that people or scientists have observed, firmly founded on observation, on what they see, what they hear, what they feel, and experiment, which continues on to be investigated. It will be continuously reviewed by scientists globally. So it's a theory which we make today, Abraham, can be disproved in 10 years' time. Abraham, the scientist, says, no, guys, you said that the earth was round. No. The earth is not round. But for his new theory to be accepted, he needs to give us firm evidence. And this cannot be just accepted by Abraham and his friends, but globally, all the scientists. You get that, Abraham?
I get that.
Good. So that is a theory. Evolution. That's what we're talking about now. And for once, Belinda, I will pick up my cell phone, which is off, I guarantee. I have a phone in my hand. If somebody told me, Abraham, when I was in grade 12, and judging by the color of my hair, that must be a long time ago. If somebody told me that I could carry a telephone this small in my hand, with which I could communicate for free nogal to all people all over the world, I would have told him, you must be smoking something. So what has happened? We had those telephones, and I remember my sister studying in Pakistan, and we had to call her, expensive. And we would then call the exchange. I want a person-to-person call to certain Aisha Gori Khan. So only when she came on, then we would then start, and we start getting billing. And today, with this, it's free. So what has happened? The mode of communication has changed from then to now. What has happened? Our mode of communication has evolved. So to evolve simply means in English to change. And let's see what it says here. Evolve, to unroll, to disclose, to develop, to unravel, to change. This is what the dictionary says. The theory of evolution states that all species of living things that exist today, those things, you and I, the ant, the bug, the spider, the bird, all these organisms and all the species that exist today, and many more that are now extinct, they don't live anymore, have evolved from simple life forms which first developed more than 3 billion years ago. This is the basic premise of evolution. And that is, and to put it, this whole thing very simply, we say that this is what the scientists are saying in terms of evolution, all living organisms developed from existing organisms. Or we can say that all organisms living today have a common ancestor. Now this word ancestor we must look at differently, not the way Abraham and I will look at ancestors like our grandfathers and our great-grandfathers. But what they talk about ancestors, when evolutionists are talking of ancestor, they're talking about that one organism that was the original organism that then developed, or rather was the beginning life form, from which, I can't say that one developed because that would be incorrect, but from which branch and rebranch and rebranch and rebranch to form species that exist today. So this is what evolution tries to explain. Some terminology that we need to learn in this year, the first of which is a gene pool. Now this is not the swimming pool as suggested here in the diagram, but it's simply, remember we did genotype and phenotype, homozygous and heterozygous, all the genes of a particular species within that population is known as the gene pool. That means the genes of all the organisms in a population. Obviously these organisms must be of the same species in that gene pool there. That brings us to another word, species. What is a species? A species is a group of organisms which share similar characteristics and can interbreed. They can interbreed among themselves, but not just, it doesn't end there. There's a third important component. And what is that component? To produce fertile offspring. So one is that the fact that they can reproduce, right? But when they have offspring, their offspring can also reproduce. That's what fertile means. They're not shooting blanks. They're shooting live ammo. They can have children. Okay? That leads us to another word, and that is the word population. What is a population? A population is a group of organisms of the same species that can interbreed. And if they are the same species, that means they can produce fertile offspring as well, in a specific habitat at a specific time. This is important. We cannot talk of a population of donkeys and zebras. Donkeys are donkeys, that's one species. Zebras are a different species. So when we talk about a population, it must be either of giraffes or donkeys or zebras. At the same time, that's the same species. Next, we cannot talk about the donkeys and zebras, full stop. We must specify the habitat. In Azadville, in Krugersdorp, in Randburg, at a specific time, in July, in August, in September, whichever time. So even in your own garden, you may have a maize plantation in your back of your garden. I'm hoping it's maize and nothing else that you're planting, or radish, or potatoes, or tomatoes. You may have a population this year, this season. Next year, the same type of species will grow there. But this particular crop that grows is not the same population that you had last year. It's a different population. Why? Because time has changed. In your own town, or in my own town, I may be living in one area, and I've got my plantation of mealies in my garden. And down at the school, we have another plantation of mealies. The plantation in my garden is a population on its own, and the one in the school is another. Although they're the same species, although they can interbreed, but they are not at the same place, so they do not form a population. Let's understand that carefully. So people, in this section, there's lots and lots and lots and lots of terminology. You need to get to grips with that terminology.
Guys, we've come to the end of today's show. thank you so much to Macmillan for making this wonderful show possible. And thank you to you guys for joining me and participating fully in today's revision lesson. Remember to visit our website, guys, for all your notes, all the videos that you need to watch from earlier on in the term. Our website is learnextra.co.za/live. Guys, if you are stuck or you need to ask any questions, please send an email to helpdesk@learnextra.co.za and let us know exactly where you're stuck so that we can help you even more. Don't forget to look out for more news on Library Month on our Facebook page as well as on our website. And guys, remember to join us for Easter Revision too. Bye guys.